Welcome, it's Life Sciences, grade 11, and we're going to be working on part 2 of support and structure and transport in plants. So we've done the whole process on the roots and the stems and how the roots and the stems of a dicot are able to absorb water and the strengthening and support all the way up the stem. We're now going to look and focus on water and the transporting of that water and the dissolved mineral salts. So we look at structure and uptake of water in plants. I've chosen pink for today, so here we go. Uptake of water and mineral salts. Now remember, the main reason a plant needs water is for photosynthesis. When photosynthesis takes place in the leaves of a plant, it requires sunlight energy, carbon dioxide, and water. And what does it produce? It produces glucose and oxygen. The oxygen is released out to the stomata into the atmosphere during the day only. Because remember, photosynthesis only occurs when the sun shines. You did this in grade 10. Please, if you can't remember, just go back and have a look at photosynthesis. So photosynthesis is a process whereby plants that contain chlorophyll, all right, use radiant energy, so energy from the sun, radiant energy, and water from the soil, okay, and carbon dioxide from the atmosphere, and it combines that to form glucose, and oxygen is released as a waste product. All right, so we need plants. First, they use our carbon dioxide, and they release oxygen. And we eat the food because plants, remember, are producers. They're producers in the food pyramid. Now, without water, there can be no photosynthesis. Why? Because we take the H, the 2 and the O, all right? And we take the C from the carbon dioxide, and that's what makes the glucose, the carbohydrates. Carbon, hydrogen, and oxygen. And your hydrogen to oxygen ratio is always 2 to 1. So we must have the carbon dioxide and we must have the H2O from the water to make the glucose. And the oxygen here from the carbon dioxide is released as the oxygen into our atmosphere. Without water, there is no photosynthesis. So the plants cannot produce organic nutrients, okay, like glucose, lipids, and fats. Okay, also remember, glucose is made of carbohydrates, which is carbon, hydrogen, and oxygen, because stored glucose is starch. Lipids are made of carbon, hydrogen, and oxygen. Proteins are made up of carbon, hydrogen, oxygen, and nitrogen. So the plants will take the nitrogen that they absorb and obtain, and they will add that to make proteins. So everything a plant needs is made during photosynthesis. All right, plants need to transport the water and the dissolved mineral salts from the roots to the leaves, and this happens via the xylem. Okay? Nutrients produced in the leaves must be translocated. Okay, trans means to move, located to another location. So the word translocated means to move from one place to another. You must know translocation to all parts of the plant via the phloem. So the water is carried by the xylem together with the dissolved mineral salts up the plant, and the phloem is going to carry the nutrients that have been produced by the leaves. They are translocated via the phloem up and down the plants till they are stored in either the roots, the stem, or the leaves, or in all three. Okay, excess nutrients are stored in the leaves, the stems, and the roots. Now, movement of water through a plant. We look at 3 steps to get the water up the plant. We have root pressure, clearly that's in the roots. Then we have water moving a little bit up the xylem, or to a point in the xylem, that's called capillarity. And then we have the suction force of transpiration. And this transpiration pull, or suction force of transpiration, this is the most important. Okay, but root pressure in the roots, capillarity in the xylem, and transpiration pull because of the leaves, those are the 3 steps to get water up a plant. Okay, that's how we get the water up the plant. All right, now a diagram to show root pressure. We had, and we did this previously, we have our root hair with the increased surface area, all right? It has a cell membrane, and then it has a vacuole. So it has a thin cell wall, a very large vacuole, okay, nucleus, and you've got your soil particles here. All right, and then from here we're going to have our cortex cells. Okay, we're not really interested in the cortex cells. What we're interested in is the epidermal cells. So there's the large vacuole there with a nucleus. This is the neighboring cell. Okay, this is going to be number 1, a thin cell wall. Number 2, a large vacuole. Number 3, no waxy cuticle. We've done this, it's just a repeat. Number 4, increased surface area. Okay, now remember, in this vacuole we're going to have cell sap. And the cell sap is made up of water and dissolved minerals. So the water is now going to move, let's do water in orange or white. Okay, the water is here. The water sitting here in the soil is called capillary water. That's the water we absorb, or at least we are going to absorb here into our root. So this water will move from a higher water potential, zhoops, through the cell wall, into the vacuole filled with cell sap to a lower water potential. The water then moves along, and it then goes either along the cell walls, or that water is going to go through the air spaces, or the water is going to go all the way through the cells. If it goes through the cells, it's going to be osmosis. If it passes around the cells, we call that diffusion. Or if it's osmosis and it moves through the cell, it's symplastic movement. Okay? If it's by diffusion, all right, it's a totally different process because it's not moving through the membrane. It's diffusion, and that would be apoplastic movement. But symplastic, it moves cell to cell. Apoplastic, it moves along the cell walls and through the intercellular air spaces. So that is how the water is going to move. Now, there will always be more water in the soil than in the root. Even when the soil is dry, the water cannot move out of the root hair, ever. The water will always only ever move into the cell. If there isn't water that can move into the cell of the root hair, then the plant will slowly but surely wilt and die. Okay, so water always moves in one direction, and this water keeps on going until it gets to the xylem vessels. So it's got a whole bunch of tissue it's going to move through first, through the cortex, through the endodermis, through the pericycle, and it will eventually get to the xylem vessels. And the xylem vessels, zhoops, in it goes, and that is going to transport the water up. So up into the xylem vessels, this is what we call root pressure. And root pressure is created because of osmosis, movement from a higher water potential to a lower water potential, along the concentration gradient and through a partially permeable membrane or selectively permeable membrane. So it'll be the cell membrane of the root hair, which runs all the way along here, all the way around the cell. Okay, you're going to have your cell membrane, okay, through the thin cell wall and through the tonoplast of the vacuole, which is the membrane around the vacuole, if you remember, and from a higher to a lower water potential. That is root pressure. Now, here it is in words. Water moves, or water is drawn from the soil, okay? The water potential in the soil is higher than in the root, so water moves by endosmosis. Now remember, osmosis, water from a higher water potential to a lower water potential along the concentration gradient until it moves through a partially permeable membrane. So it's always from a high to a low. If it moves into the cell, it's endosmosis. If it moves out, it's exosmosis. So endosmosis, the root hair now has a higher water potential relative to the adjacent cells. So the water moves from the root hair to the cortex parenchyma, past the endodermis, through the Casparian strips. Just a reminder, the Casparian strips of the endodermis, the cells look like this. And they've got the Casparian strip all the way around here. So it channels the water in here and out here to the xylem vessels. Those Casparian strips, people, remember, very important. And then into the vascular stele. The vascular stele is where the xylem is. Okay. If it moves, and I told you from cell to cell, just think cell to cell, it's symplastic pathway. And if it moves around and through and along the cell walls, but never goes through the membrane, then it's apoplastic. So if it goes through the cell membrane, then it's osmosis. If it moves around the cell wall, it hasn't gone through a membrane, it is diffusion. So apoplastic is diffusion, and symplastic would be osmosis. Some water moves intracellularly, okay, but most of the water is going to move intercellularly via the air spaces. If it is intra, it is inside. Inter means between, okay. A high water potential develops in the root xylem because of the force of root pressure, okay, because of the force of root pressure. Let's just recap. Water goes in from a higher water potential to a lower water potential through the thin cell wall of the root hair, okay, through the cell membrane, so that's osmosis, okay, through the cell membrane, which is semi or selectively or partially permeable, into the cytoplasm, and then in through the tonoplast, which is the membrane around the vacuole. The water then moves from the root hair into the neighboring parenchyma cells, okay? So from a higher water potential now to a lower water potential in those parenchyma cells. And then it'll go from cell to cell, then it is symplastic pathway by osmosis. Or it moves around the cell walls, apoplastic pathway, or it moves through the intercellular air spaces. So the water moves along from cell to cell, from a higher to a lower water potential until it gets, or a higher to a lower water concentration if it's diffusion, until it gets to the endodermis. At the endodermis, we've got the Casparian strips with a nice strip around the cell, directing, they're also called passage cells, by the way. And it passes the water, channels it through into the xylem vessel, so it can be transported up the plant. And that water moving from a higher water potential to a lower water potential, or higher concentration to a lower concentration if it's diffusion, is going to be what we call the force of root pressure. Okay, structural suitability of root hairs. Now, how are they structurally suited? Now, people, there are two really important components that you need to know, especially with regard to cells. First of all, what it looks like, its structure, relates to what it does, function. If a cell looks a specific way, it has a specific function. Okay? If it has a thin cell wall, well, guess what? Diffusion is going to be taking place into that cell. In the root, the root and the epidermal cells didn't have a waxy cuticle. They didn't need one. They want water to come in. If there was a waxy cuticle, it'd be like putting a raincoat on. Okay, and then going and standing in the shower. The whole idea to be in a shower is to wash yourself. If you put a raincoat on, you can't wash yourself. The same thing here. So no waxy cuticle in the stem. There was a cuticle. Why? We don't want water loss. Structure versus function. Now, the structural suitability of the root hair, what does it look like in order to perform what function? Let's look. You must know this. If I was setting your paper, I would ask this to you for 4 marks. Or 8 if you had to put the reason as well. So, root hairs have an increased surface area for efficient absorption. Remember, instead of the root hair looking like that, the root hair looks like that, but it's got this long piece that sticks out over there. So you've increased this surface area by all of that area there. Have a thin permeable cell wall. Why? To allow unrestricted movement of water into the structure. Have a large vacuole. Remember, large vacuole means a large tonoplast and also a lot of cell sap to maintain the concentration gradient. In other words, from a higher water potential in the soil to a lower water potential in the root hair. This sign here is your water potential sign. Okay, and there is no waxy cuticle present. You must know these 4 points, please. Okay, question 1. The following diagram represents part of a cross section through an angiosperm root. In other words, a flowering plant, therefore it is a dicot plant. Okay, what are we going to have? First thing you do, label the diagram. So, this here is my root hair. Okay, then those are my cortex cells. And they're made of parenchyma. All right, number 3, look, you can see the little black spots there, which means it is the endodermis. 4 is the pericycle. Okay, and 5 is the xylem. And just so we remember, these black strips that we have around the epidermal cells are the Casparian strips. Okay, Casparian strips. Now, explain how water enters structure numbered 1. So we're going to have to explain that whole process. So we say, firstly, let's get a different color. First, we're going to have the soil particles and the capillary water. Okay, and we're going to have a higher water potential in the soil compared to a lower water potential in the vacuole. So water is going to move from a higher water potential through the thin cell wall into the cytoplasm of the root hair, through the tonoplast, okay, so it's gone through the tonoplast and into the root hair. From the root hair, the water is going to move into the cortex cells, all right? Now, the water can either move through the cortex cells, and if it's going from cell to cell, it's symplastic movement of the water, or the water will move via the cell walls and the intercellular air spaces, okay, all the way along until it gets to the endodermis. When the water gets to the we only had to stop there. We had to go to here. Sorry. So, water moves from a higher water potential to a lower water potential in the root hair, through the cell wall, thin cell wall, through the cell membrane, into the tonoplast, and into the vacuole, from a higher to a lower water potential by end osmosis. All right. And that would be your 7 marks. Sorry, all of this here is extra. Then, write down the names and numbers of the tissue through which water passes until it is ready to be pulled up the plant. Okay, so that's what I carried on doing just now. So we're going to use this space here, and we're going to say we're going to go number 2, which is the cortex cells, number 3, which is the endodermis. So we're going to go, this is the answer to question 1.2, because I don't have any space. We're going to say this. We're going to say it's going to go through number 2, the cortex cells, and then number 3 was the endodermis with Casparian strips. Okay, let's just have a look at our diagram again. And then number 4, the pericycle, and then number 5, it's going to get to the xylem vessel. So number 4 was the pericycle, and number 5 was the xylem vessels. Okay, so there are your 4 marks. So 1, 2, 3, and 4. And if you didn't put the numbers, you would have only got half marks. Okay, but they wanted the numbers. You've got to read your questions carefully, and it says write down the names and the numbers. All right, then 1.3. Name the force which develops in the root to move water from the soil to the central stele. In other words, to where the xylem is. And that force is root pressure. The water keeps on moving into that root. Name 4 ways in which the root hair is structurally suited to its function. I'm not going to write this out. We just go back our slide. It says root hairs have an increased surface area for efficient absorption. They have a thin permeable cell wall to allow unrestricted movement into the structure. They have a large vacuole to maintain the concentration gradient from a high to a low. And then they have no waxy cuticle. Now we move on to our next section. We've done root pressure. Now we move on to capillarity. And capillarity is water as it gets into the xylem. So water enters the root xylem and passes through the xylem vessels and tracheids. Because remember, xylem is made up of vessels and also tracheids. Okay, the force called capillarity takes place in the xylem. Now, it consists of cohesion and adhesion, which I'm going to explain to you now. Cohesion is a force where water molecules stick to each other. They're cohesive. It sticks. And adhesion, it sticks to the sides. All right. So in English, we'll say that the class, because all of you are friends and you stick together, we'll say that you form a cohesive force. Okay. Cohesive unions form a cohesive force. Stick to each other. Adhesion, stick to the sides. If I want something to stick to something else, I use an adhesion glue or an adhesive glue. All right, so cohesion, stick to each other. Adhesion, water molecules stick to the walls of the narrow vessels and tracheids. Both forces together ensure that water moves up a portion of the stem. Now, there's an experiment that we used to do in the old syllabus. We used a trough of water, and we used quite a wide tube. And then we used a narrower tube. And then we used a very thin tube. And we put that in a trough with water. And you can try this at home as well, or when you go out. And what would happen is, in a really wide tube, you'd have this. The water level would be pretty much what the water level is on the outside in the trough. Okay, so you'd have the water level to there. When you put it in a narrower tube, the water level would rise a little bit. There's your meniscus, and the water level would rise just a little bit. And you put in a very thin tube, and the water level actually goes quite high up, okay, in that tube. So the thinner the tube, the higher the water will climb. And this is an experiment to show capillarity. Now, what I'd like you to do is take a wider structure. You know, some places you can actually buy, or you are given really wide straws, especially if you're sucking like slush puppy. So the next time you go to movies and you have a slush, one of those slush cups, keep your straw. It's quite a wide straw. And then if you go anywhere else and you get a normal thin straw like the size of a pen, take those two straws and go home and take colored, put some food coloring in water, or you can even do it with flat Coke. Okay, Coke, you'd be able to see it nicely. And you take your fat straw and you shake it around, and you put your thin straw in and you shake it around and have a look. The level in the fat straw will be a little higher than the level of the Coke or the whatever fluid you're using. And if you use coffee or tea, and if you're thinner the straw is, the higher it's going to climb in the tube. That, people, is what capillarity is all about. It is the cohesion, the water molecules sticking to each other. That's cohesion, force. And your adhesion force is why water has a meniscus like that. So whenever you see the top surface of the water, it will always look like it's climbing up the sides. And that is an example, or it shows the adhesion force. So when they say here the force called capillarity, it's made up of cohesion and adhesion. All right. Cohesion, water molecules stick to each other. Adhesion, the water molecules stick to a thin tube. So the narrower that tube, the higher those water molecules are going to climb. Another way to remember it is if you, for example, if I give you a big wide cylinder and I say you've got to climb to the top, you're never going to be able to, because you can't jump anywhere. It's smooth. The wall is round. It's smooth. You're going to be at the bottom. But if I give you a tube that's this big, you'll be able to climb up it. Why? Because you'll put your back on the one side, your feet on the other side, and you'll be able to climb up it. And if there were a lot of people in there, we could actually push each other up because we're sticking together. So that is what happens. Capillarity in the xylem. So we have root pressure in the root, capillarity in the xylem, and that gets the water sort of just above soil level. All right? And then we look at our third most important force, which is transpiration pull. But let's look at our xylem vessel. Okay? Xylem vessel I drew just now, but it's this. You have this thick cell wall, and it's waterproof. So you have this waterproof, thick cell wall. Okay? And it's just one long tube. All right? It's rounded, so it's strong. Okay? This wall is thickened with lignin. Okay? It has a large lumen. A lumen is this inside cavity. There are no cross walls. All right, people, remember that your xylem cells started off like this. They were cells that had cross walls. These are cross walls. Okay, that's a cross wall, that's a cross wall. And the cell walls started to thicken. And the thicker these cell walls got, okay, the less nutrients and water and everything else these cells on the inside had. Eventually, the cells on the inside die. And what happens is the cross wall starts to disintegrate. And then the cross walls disintegrate properly until we end up with this long, narrow tube. So there are no cross walls. The cross walls have disintegrated. Just this one long tube. And it's rounded. Okay, it's rounded for strength. Okay, but now because there are no cross walls and because this thing is thickened with lignin and it's waterproof, we need to make sure that we can get liquid. If this poor xylem vessel is full, it's got to be able to pass that liquid onto the one next to it. So we'd have another xylem vessel here and another one there. And how do we do this? The xylem vessel has little structures called bordered pits. And those bordered pits are holes, and the holes will then pass liquid. If this is another xylem vessel here, and it's got a hole there, the water will come in here, okay, or it will be on its way up, sorry. But as it's passing up here, it may pass out here and into there and then go up like that. So it allows for cross movement of the water up the xylem vessels. The most important thing, though, is that those xylem vessels, those cell walls, are thickened with lignin, and they are non-living cells. There's nothing living in here anymore. Look here, all the living contents have been washed away, so they are non-living. And they are long, narrow tubes. All right, so we look at the structural suitability. In other words, how is the structure suitable to the function of the xylem of carrying water up the plant? First of all, they are non-living because everything's been washed out. Okay, elongated, the long, that help rapid movement of water. The tracheids overlap each other. Water is conducted from one cell to another by pits, okay, that link the cells. And they're no longer cells, they're just tubes. Okay, cell walls are thickened with lignin, which helps to withstand the pressure of adhesion against the cell walls and cohesion, the molecules sticking to each other. Okay, cell walls are perforated and with lots and lots of bordered pits, and that allows water to move sideways. Okay, they have large lumens and no cross walls, which allows for unrestricted flow of water. Okay, there are no cross walls. They don't have to do any little jiggle jaggles. They just go straight up. It's like a big fat straw that you're sucking liquid up. Okay, vessels and tracheids are cylindrical. They're rounded, which makes them structurally strong. In our previous session on structure and support of plant tissue, I said to you that if a cell is round, it's much stronger than a cell that's square. Okay, so square is fine if you're going to have them on the outside and they're there to form a layer to protect. But when they are rounded, there's a lot more strength to be able to carry this force of water. And water is probably the most, it's the strongest force we have on this earth, if you look at what water can do. All right, moving on. You have to know experiments. So this experiment is going to be to demonstrate the movement of water through the xylem. So how do we do it? We take our plant, we grow a plant, we remove it. We don't take it and rip it out of the soil. We take it and we gently loosen the soil around the plant, and then we rinse it very carefully and very gently in a bowl of water. Once you've rinsed all the soil off, we take our plant, roots and all, and we stick it in a beaker. So this is a beaker. This thing here is a retort stand. Okay. Retort stand, that's a clamp. Okay, there's our plant. Those are the leaves. All right, and then we put in eosin solution. Now, eosin, they also use in food coloring. It makes red food coloring. So you can use red food coloring, you can use eosin, and you put it in here. Okay, you can also use mercurochrome, by the way. And you put it in here, in this water. And you leave this plant for 2 or 3 days. Clearly you put it where it's nice and sunny and it gets lots of sunlight and it's a happy little plant. It doesn't even know it's out of the soil. Okay, leave it in the eosin or mercurochrome or red food coloring, all the same pink stuff, and it then absorbs into the root. All right, into the root and into the xylem vessels. Okay, and if you leave it for long enough, you'll actually see that the leaves, the main veins, start to discolor and go like a reddy color, or as red as you can get, green and red. Okay, and that will then prove transpiration. But something else you do then is you'll see it in the leaves if you leave it long enough. But if this is all eosin solution, it goes in through the roots, up the xylem, and into the leaves. And you'll see the discoloration of the leaves, okay, of the veins only, not of the actual mesophyll tissue. But what you also do then is you can take this poor plant out, show me mutilating it, and then you take the stem and you cut through the stem like that. You take a very, very sharp scalpel and you go zips and you cut it. And if you look at the stem will look like this. And you'll actually see where the xylem is. You won't be able to see the phloem, because remember the phloem would be there. You won't be able to see the phloem or the cambium or anything else. You'll only be able to see where the xylem vessels are. And that is an example experiment to demonstrate the movement of water through the xylem. Why? Because you can see that it's the xylem carrying the water up the plant. Okay, transpiration pull. This is our third force, and it is the most important to get water up a plant. It's the one that works the hardest. Okay, transpiration pull is the loss of water vapor through the stomata of the leaves during daylight. That is transpiration, and it causes transpiration pull, or we say the suction force of transpiration. So transpiration is the loss of water through the leaves of a plant. Specific structures are the stomata, where water moves from a higher to a lower water potential from cell to cell until it gets to the substomatal airspace, and there it diffuses into the substomatal airspace and out of the leaf. Okay, this force is responsible for sucking the water all the way up the stem to the leaves. It is the most powerful of all three forces. You've got to know that. All right, so if we look at transpiration pull, what I did here is I did a little schematic for you. So you have the terminal ends of the xylem vessels in the veins of the leaf. Okay, the water then moves through the spongy parenchyma, into the intercellular air spaces, into the substomatal air chamber, and out to the stomata in the form of water vapor. Okay, it's water vapor because it's in gas form. All right, so we have the terminal ends of the vein, the xylem in the vein of the leaf, middle part, the veins of the leaf. Then we have the spongy, it moves through spongy parenchyma, the intercellular air spaces, the substomatal chamber, that's before if the stomata are here. And then you've got your epidermis. Your substomatal chamber is here. There, that's the substomatal chamber, and this is outside, so the water moves out to the stoma, which is the pore, okay, in the form of water vapor. So here we have our leaf, and you don't need to learn the whole mesophyll of the leaf again. You did that in grade 10. What you have to be able to do here is the path of water from the xylem of the leaf. So what do we have? We've got our xylem vessels. You can see, well, you can't really because this isn't a very good diagram. It didn't come out very nicely here. But there are your xylem vessels with their nice thick cell walls, okay, and the large lumen, etc. Okay. And the water then comes from up the stem and into the leaf. Now remember, the xylem vessels are on the inside, okay, as they're coming up the stem, and then they go into the leaf. So the xylem is always on top. The phloem will always be at the bottom. So here, if you think of the stem, if this is a xylem, a vascular bundle in the stem, this is the xylem, this is the phloem. So here it's coming up the stem, and now it goes into the leaf. The xylem's on top, the phloem's on the bottom. Okay, so xylem vessel, the water will then move from cell to cell. If it's going from cell to cell, it's moving by osmosis. If it's moving along and into the intercellular air spaces and along the cell walls, etc., and then into this substomatal airspace, then it's moving by diffusion. The water then diffuses from these mesophyll parenchyma cells, okay? That's diffusion into the substomatal airspace, and then it moves out through the stoma, which is the pore, in the form of water vapor. People, please remember water vapor. Alrighty, now, when the water exits the leaf, the water potential is changed, resulting in a negative pressure developing in the xylem. So in other words, we've gone, the water's moving up the xylem, the water then moves cell to cell, from a higher to a lower water potential if it's moving by osmosis, from a higher concentration to a lower concentration if it's moving by diffusion. Diffusion, you always talk about concentrations. Osmosis is always water potential. Okay, so we have a suction, I'm going to change colors here. We have a suction force is transferred along the leaves and the stem, causing a decrease in the water potential of the roots. Why? Now think about it. What's happened? Water comes from the soil into the root hair to the xylem. Then it gets carried in the root xylem up until it's just above soil surface, about 1 meter, 2 meters above soil surface, okay, if it's a really big tree. Then that's by capillarity. So root pressure, capillarity. And now this water has to go out of the leaves that are 20 meters or 10 meters high. So we've got to get this water there. So it creates this suction force. So as one water molecule moves out there, this can move higher. Then this water molecule, another water molecule moves out, and the next lot. It's like a queue. If you can imagine a queue of people when you want to go to movies, for example, or when people are going to vote. There we go. When people are voting, you've got this long queue of people. People go in, okay, they go and they vote, and as they come out, the next person's allowed in, and then the next person, and the next person. And that's exactly what you're doing. You're creating, as this person leaves, there's a space. So there's a lack of, there's a vacancy. So one person can move in, and everyone shuffles closer in the queue. Now, if you can imagine that, that's exactly what they're talking about here. We have a negative force that is created because we have a decrease in the water potential. All right, now, transpiration assists to regulate the concentration of the cell sap inside the plant. Okay, now you must remember, a plant's got a certain amount of water, and that depends on how much water is in the roots. Okay, so if it's a really hot day, that plant's going to be transpiring a lot. Why? Transpiration is not sweating. Please don't confuse transpiration with sweating. What happens is you've got your leaf. The water comes out of the leaf. As that water vapor comes out, and if there's wind or it's very hot, it evaporates quickly. So now there's a space. So now the next water vapor drop of water vapor can come out, and it goes away. And another one, and it moves away. It evaporates. So what's happening? As one moves, another one has to move to take its place. So the plant starts dehydrating when it is very, very hot because of transpiration. So one of the questions you could get in an essay type question is, some scientists have said that transpiration is a necessary evil. Explain. Now, transpiration is necessary, and it's an evil because too much transpiration and not enough water coming in through the root can actually damage the plant because the leaves will wilt. It loses turgidity, starts to go all blur, okay, until we give it water, and then it starts sprucing up again. So it is a necessary evil, but why do we need it? Because if the leaves don't have water, they cannot photosynthesize. They cannot produce food for the plant to stay alive. Okay, so transpiration assists to regulate the concentration of the cell sap inside the plant. Plant cell walls are made of cellulose, providing structure to the plant. Transpiration can cause excess water loss. That's our necessary evil, where the plant will wilt and may even dehydrate completely and die. Okay, this is a necessary evil. Okay, when a plant has sufficient water, in other words, got enough water, the vacuoles fill with cell sap, creating pressure from the inside called turgor pressure. Now let me just show you what turgor pressure is. If I have a cell, it's a cell wall. Okay, then we have a membrane that runs all the way right next to the cell wall. So there's the cell membrane. Let's do it like this. Okay, we have a vacuole. Now that would be the tonoplast. That's the membrane around the vacuole. This is the vacuole with cell sap. And you'd have your nucleus. And then clearly this is your cytoplasm. Okay, now turgor pressure is the pressure that's exerted in the cell. When the tonoplast is nice and this vacuole is nice and full and the cell is perfect, it means that the pressure on the contents of the cell pushing outwards against the cell wall and the cell membrane is equal to the pressure pushing this way of water wanting to come in. And when those two pressures, the cell pressure and the turgor pressure, when they are equal, then the cell is turgid. So when the pressure from outside to put water in and the pressure from inside to push water out, when that is equal, your water molecule is going to go from here, it'll go into the cell and another water molecule will come out. And when that's equal, that's when the cell is turgid. The cell is full. And we say that that creates turgor pressure, which is this pressure that we're talking about over there. So you've got your turgor pressure provides support to herbaceous plants. Herbaceous plants, those would be plants where they have green stems. Okay, there's no secondary thickening. These are plants that will only live for one season, generally. All right, they may live for a year, but generally one season. So they have their stem is green. Their stem never matures to have secondary thickening. All right, when cells lose turgor pressure, they begin to droop and wilt. So in other words, when water is lost here, so we don't have this water molecule coming in anymore, the water molecules are just coming out all the time because the water potential on the outside is now lower than the water potential in here, which is higher, then the water will move out of the cell. And when that happens, the cell will therefore wilt. And if it wilts too much, it will eventually die. Okay, demonstration of transpiration of water through leaves. This is a demonstration, people. All right, that's all. It's a demonstration. Now, I actually took this from one of the old textbooks, and it's a very nice diagram. So here we go. Experiment and the control. Why do we have a control? Well, otherwise we could say we did this, and it was because we did this that happened. We are trying to prove that transpiration occurs to the leaves. So we can put a leafy twig and say, look, transpiration occurred. Well, how do we know for sure it was the leaves that transpired? It could have been the stems. We don't know. So we have to do a control to show that it was actually the leaves that water transpired through. So we're going to have our beaker, and we're going to put a layer of oil across the top of the water. Why? Because water can't evaporate through the oil, which means it's not the water from the beakers that collected in the bell jar or the plastic bag. You can use a plastic bag instead of the bell jar if you don't have equipment. But then it's not water from this beaker that caused it. So that's why we put the oil there, to take a precaution against any water from the beaker evaporating and collecting in the jar or plastic bag. If this, you can also, I'm going to put your bell jar or plastic bag. So there's no excuse not to do this experiment. It's a very easy experiment. You don't even have to use a beaker. You can use a plastic cool drink bottle that you cut. That's makes a perfect beaker. And then you just need water with a little bit of oil, but you first put your plant in and then you put your oil. Otherwise, you're going to put oil all around the roots. So first the beaker, you have your beaker with water, put the plant in, or the piece of twig, and then you put your oil on. Okay, this is a leafy twig, or twig with leaves. You have, if you're using a bell jar, you'll use a glass plate with petroleum jelly. If you don't have that, it's fine. Use your little plastic cut-off water, a cool drink bottle with your plant, and just when you put your plastic bag all the way around the plant. Okay? And then you have cobalt chloride paper, which tests for water. Okay, but you don't actually need that. What you do need is, you'll see if you just use a plastic bag, water droplets will form on the inside. Where there's just a piece of leafless twig, very little water, far less water. There'll be tiny little bit of water, but not a lot. Why? Because remember, this is a leafless twig, but it's a twig nevertheless. And the end is still going to be greenish. If it's greenish, it's going to have stomata and chlorophyll. So that you'll have your leafless twig. But your cobalt chloride is to test if there's water. It goes pink if there's water and blue if there isn't. It's the other way around. I mean, water's blue. So just remember, it's opposite. Okay, how the structure of the leaves affects the rate of transpiration. People, the number of stomata. Now think about this. If there are lots of stomata, the leaf is going to do a lot of transpiration. Why? Because there are all these openings for water to come out of the leaf. Okay? More stomata, let me write it here. More stomata equals more water released. It's not lost, it's released into the atmosphere. Okay? And remember, your stomata are mainly found on the bottom surface of a leaf, not the top. Waxy cuticle, well, if it is thicker, less transpiration. And all you have to do here is think of xerophytes. Xerophytes are your plants that live in a desert. Okay, they're going to have a really thick cuticle so that they can survive in areas where it's very, very dry. They don't lose a lot of water through their leaves. Okay, otherwise they'd be all wilted and shriveled up. They're not. Think of your succulent plants. They have nice fat, swollen leaves. So they keep their water in. Why? Nice, thick, waxy cuticle. The thicker that cuticle, the less water loss. The thinner the cuticle, only a little bit of water loss. And if there's no cuticle, it's going to just go and wilt. All right, hairs. Now, think about the surface of the leaf. If we've got this leaf surface like this, okay, and it's got these little hairs all over, like an African violet. All right, as the water, and also underneath the leaf, as the water comes out, the water is going to stick and be trapped by those little leaves, which will make the surface around the leaf very moist. So it will slow down the rate of transpiration. So the hairs, the more little hairs there are, the less water is always going to just go off, because the water will stick to the hairs and make it moist around the stomatal opening. So less water will pass to the atmosphere. The shape and size of a leaf. Oh, people, please, just you can use your common sense here. If we look at a pine needle, it's a thin thing like that. There's going to be a lot less water lost in a pine needle, so if it's thin, than in a nice big fat broad leaf like that. Okay, a big fat leaf like this has got all this surface area to lose water from, whereas a thin stringly pine needle or a leaf that's long and thin like that, it's never going to lose as much water as a big fat leaf. And if you think of plants that you're going to find in areas where it's lots of water, like at the equator, you're going to have plants that have huge fat leaves like this so that they can lose as much water because they're absorbing water at a higher rate. So they've got to get rid of all the extra water. Whereas if you go to very dry places, you're going to find pine trees. Why? Because pine trees don't need that much water, but also because they can survive in the cold. So if you go to places where there's a lot of snow, your pine needles are fine. That snow is not water, it's solid. But they survive there. Why? Because they lose so little water from the surface area of their leaves. So it's the shape and size of the leaves. Just think of surface area. Okay? The arrangement of the leaves. Now this is so clever. Plants that live in areas that are very dry, the leaves will arrange themselves so that these leaves are shaded by the ones just above them. And they are shaded by those, and they are shaded by those. So it's only really the leaves that are right on the outside of the canopy that are going to be quite dehydrated. The rest of the leaves are shaded by other leaves, so they don't lose as much water. They stay in the shade. Now, if I hang a t-shirt up in the shade versus in the sun, which one's going to dry quicker? Clearly the one that's in the sun. So they shade, so it's the arrangement of the leaves. And then water retaining capacity of the leaves, like for example in succulents. Your succulents are plants that grow in very dry areas and they have big fatty oily leaves. Think of aloes. They've got really scrunchy leaves. And there's a lot of liquid that stays in the leaf because of this thick waxy cuticle, so they can retain water in the leaves. Okay, we now look at we almost finished, people. We look at environmental factors. In other words, factors that are outside the plant. That we've looked at everything from inside the plant. Now we're going to look at outside the plant. And we say, okay, what environmental factors here are going to affect the way a plant grows? Well, think about it. Temperature. If it's a high temperature, then we're going to end up with lots of transpiration. Okay? If the temperature is low, a plant's not going to transpire as much. Again, think of that t-shirt that I've just washed that I want to dry. If I dry it where it's very hot, and I chuck it in a tumble dryer, it dries very quickly. If I go and I take that t-shirt and I hang it in the shade where there is less heat, it's going to take a long time to dry. Those are environmental factors that will affect the rate of transpiration. It's the rate at which transpiration takes place. Transpiration will take place, but how quickly or how slowly is the rate at which it takes place. Humidity. My same t-shirt, if I want to dry my t-shirt, is it going to dry better if I hang it on the washing line outside, or if I take it and I stick it and I hang it up in the bathroom where I've just had a shower? Where do you think it's going to dry quicker? Well, definitely outside, where there is less water in the air. If I do it in the shower, where I've just had a shower and the air's all wet and moist, it's going to take a long time for that t-shirt to dry. So, humidity. Humidity is the water in the air. Now think of the free state, where it's very dry, and also in Gauteng, where it's very dry, in most of the regions of Gauteng, versus KZN, Durban. Durban, the air is moist. As you come over the hill and you start heading down past Pietermaritzburg towards Durban, all you have to do is roll your window down and you can actually feel the air getting thicker and wetter as you get to Durban. So I love Durban. Okay? If you look at the people in Durban, their skins are always beautiful because they don't dehydrate like ours does here in Gauteng or in the Free State. All right? In Port Elizabeth, it's near the coast, it should be wet, but there's always wind, and the same in Cape. In Western Cape, the wind blows all the time. So the wind will blow that humidity away, but it's still more moist there the air than it is here in Gauteng or in the Free State or in Limpopo or in Mpumalanga. We have very dry areas, okay, very little humidity in the air. When it's rained and you walk outside and that wonderful smell that you get, and it's wet, the air is heavy because it's moist. So it's humidity. Humidity is the water in the air. And if there's lots of water, that leaf is not going to transpire as much. Why? Because the water potential or the water concentration in the leaf is higher than the water concentration outside, or it's about the same, depending on how much water there is or how much humidity there is. So the water battles to come out of the leaf, so it stays moist in the leaf. It slows down the rate of transpiration. So for humidity, if there is high humidity, it means that we are going to then have slower rate of transpiration. It's much slower. Wind, common sense will tell you, water comes out of the leaf and the next thing the wind blows it away. So there's going to be a gap there. So the next water molecule can come out. So the more wind there is, the higher the rate of water transpiration. So lots of wind will equal high transpiration rate, because the water will be moved away from the leaf surface quickly. If there's less wind, well then, or there's no wind, the water's not going to move out. Think about your t-shirt that we're trying to dry, okay? If I hang it in the wind, it's going to get dry very quickly. If I hang it in a place where there isn't any wind, it's going to dry slower in comparison. And then light. Now, light, very important, because the more light we have equals open stomata. The less light you have, the less the stomata are going to be open. Why? Because remember in grade 10, the stomata photosynthesize. And the more sunlight they have, the more photosynthesis takes place, the more glucose they produce, the lower the water potential in the stomata to open. Okay, so it will then absorb water, get nice and full, and open nicely. So we must have light. More light equals very open stomata. Less light, the stomata start to close. At night, the stomata are closed completely because there is no light. Availability of water. Now, people, you just have to think. If there is no water coming in from the soil, so it's very dry, and there is no water, it's like when you forget to water the pot plant in your bedroom, okay, or the pot plant in the kitchen window. It always looks so pretty in the kitchen window, but they're going to be replaced like every 2 weeks because everyone forgets to water them, okay? But if that plant, the roots, have no water to bring into the plant, and it keeps losing water over here, eventually there isn't enough water in the plant to pull up into the leaves, and the plant will then die. It will wilt and die. So there must be lots of water available in at the root side. If there isn't for the roots, eventually the water runs out and the plant will die. Okay, this experiment to demonstrate the effect of environmental factors on the rate of transpiration. Now, if I wanted to look at wind, I would use a fan. If I wanted to do light, I'd use a special light bulb. Okay, move it closer or further. If I wanted to do humidity, I'd put a bag around here, okay, with a big blob of wet cotton wool, okay, to make it moist. All these things you would do to this setup to change the experiment, to see the rate of light, or how light affects the rate of transpiration, how humidity affects the rate of transpiration, how wind affects the rate of transpiration, etc. This apparatus is called a potometer. It's a potometer. And what we do is this end of this structure, this is the potometer. sits in a beaker of water, so it can always absorb water from here. It sucks the water up there. You put a little air bubble in here. You get the air bubble by lifting this end up, and then the air bubble will be sucked to here where you've got little measures. It's like a little ruler here. And this would be 1, and this would be 10. Okay? This is a reservoir. And what this reservoir does is, as the water bubble moves here, now I want to push the water bubble back. So I open the little tap and it'll push the water bubble back to 1 again. And in that way, I can do my experiment 3 or 4 times. Here you have a leafy twig, which you cut underwater. And then you set this whole experiment up underwater, and then you lift it so you don't get air bubbles in your xylem. And that here, clearly you're going to make sure everything is nice and airtight and sealed. And then there are my leaves. And they're going to photosynthesize. Now, as those stomata open, the water is going to come out of here by transpiration. And that means it's going to suck this little water bubble this way as it sucks the water up into the xylem. Now, if I want to drop the water, I can't make the water come back in here. So you open the reservoir and you push the bubble back. So if I take a heater and I stick a heater there, and then I measure how quickly it takes. 2 minutes for that air bubble to move from 1 to 10. Cool. And I move the heater further away, or the light bulb, or the fan. And you can measure the rate of transpiration using a potometer. All right, people, now we look at question 2. And here we have a diagram of the pathway through the xylem tissue of the leaf until it is released into the atmosphere. So clearly over here is where the atmosphere is. This is where the air is. All right, the atmosphere. Now study the diagram and answer the questions that follow. Remember, our rule is always look at the labels first. So here, we're not sure what this is, but if that's the xylem, it means that Z is probably the force that takes the water up, which would be capillarity. Okay, 1, well, 1 is the cell, but it's pointing to the arrows, so that is clearly osmosis. Now remember, when the water moves through a cell and it moves through that partially permeable membrane, then it is osmosis. If it goes around the membrane and goes along the cell wall but doesn't go through the membrane, it would be diffusion. All right, now X, clearly the leaf mesophyll. So that's the leaf mesophyll cells, okay? Sorry, fill. And then we also have this process here in the airspace. That would be diffusion. All right. And then here we've got the water moving out of the leaf. This would be diffusion as well. But what kind of diffusion it would be? And I'm going to write it here. Transpiration. Okay, that would be the process of diffusion for transpiration to occur. Now we know what we're looking at, we can move on to our questions. And it says here, name the parts marked X and Y. X is the leaf mesophyll, and Y, we forgot, Y is the, is the guard cell. Let's just fix that one there. It would be the guard cell. Okay. 2 guard cells would make the stoma. The stoma would be the hole that the air is going to move, or the water is going to move through. So X, leaf mesophyll, and Y would be the guard cell. If we look at question 2, name the processes involved in the movement of water at points 1, 2, and 3. So look, they're very careful to say here, it's the movement of the water. So at 1, it's going to be osmosis. At 2, it is diffusion, and at 3, it is diffusion. All right, one could argue that here, because the water is, if the water is moving through a cell, then you would have osmosis here as well. If the water moves around the cell, it would be diffusion. So just to be safe, you would say that 1 was osmosis. 2 could be osmosis, but it could also be diffusion. and 3 is definitely diffusion. Just to recap here, remember with osmosis, it's the movement of water from a higher water potential to a lower water potential along that concentration gradient through a partially or selectively permeable membrane. Diffusion is the movement of any substance from a higher concentration to a lower concentration along the concentration gradient until an equilibrium is reached. All right, so people, just be very careful there. If the water moves through a membrane, it is osmosis. If it moves, it doesn't move through a membrane, it would be diffusion. Also osmosis, it's a water potential, and diffusion, you talk about a concentration. All right, name 4 ways in which the xylem is adapted and structurally suited for the upward movement of water. Okay, what we're going to do here is I'm going to go to a very basic diagram of the xylem. All right, we've done this before, or we did this earlier. Okay, there's my xylem vessel. Or your vessels and your tracheads, it applies for both of them. Okay, and you've got this long tube. So number 1, they are non-living. Okay, and because they are non-living, they are also elongated. Okay, there are no cross walls. And that's what makes long tubes. And they are narrow. Okay, we have a wall here thickened with lignin, thickened cell wall, and it's thickened with lignin, which is waterproof, okay, which makes it very strong. We've got bordered pits, and they allow sideways movement of water. Okay, because remember you'll have another xylem vessel over there, and you'll have this one will also have bordered pits, so the water can move from here into there and from here into there happily because this wall is waterproof. Okay, you also are going to have your tracheads are cylindrical, so the tracheads are going to look like this, and they're rounder, and you've got the rounded edges, and which immediately makes something strong. So here, everything is cylindrical and round. There are your structures versus function. It's non-living elongated tubes to allow for movement of the liquid up the xylem. Number 2, there are no cross walls to restrict the water flow. Number 3, the cell walls are thickened with lignin, okay, for extra strengthening. Okay, bordered pits allow for sideways movement of water. People, you must learn your diagrams. I promise you now, if you learn your diagrams, and it's so much easier to learn a diagram than it is to learn a whole lot of writing. Learn your diagrams. Do a simple drawing like I did here now for the xylem vessel. And then as you put the labels in, put the functions as well. So it's structure versus function. Okay? Right, then we look at question 2.4, and that was 4 marks, quick, easy marks. Number 2.4, briefly name the force that causes movement of the water through the xylem as shown by arrow Z. So let's just check arrow Z here. We already said was capillarity. And remember, capillarity is a force that's made up of cohesion of the water molecules to each other and adhesion of the water molecules to a tube. So the thinner and narrower a tube, the higher the water will move by capillarity, or rather specifically the adhesion force. So it is capillarity. And then 2.5, list 3 environmental factors that would increase the rate of movement of water out of the leaf. In other words, I'm going to put your IE. In other words, transpiration. Okay, so transpiration is the rate at which water moves, or transpiration is the movement of water out of the leaf. The rate would be affected by, for example, now remember we spoke about a t-shirt that is wet, and we want the t-shirt to dry quicker. What would we do to the t-shirt to make it dry quicker? The same applies to the leaf. Okay, we'd put it in a high temperature. So if there was high temperature, more transpiration would take place, or an increased rate of transpiration. If we wanted to dry, there were low humidity. You wouldn't hang your t-shirt in a shower after you've had a shower, you'd hang it outside, where there isn't that much water inside this enclosed space. So the lower the humidity, the quicker the water will evaporate, or the quicker transpiration rate will be increased. So we've got that. I'm trying to think of the lot. Wind. If I wanted to dry a t-shirt quickly, I would put it in the wind. I'd put it where the wind was blowing, not in a room where there was no wind. So clearly it would dry quicker. Evaporation rate would increase if there was wind. So all told, you're looking at a high temperature, low humidity, and an increased wind factor. And all of that would then give you a higher or increased rate of transpiration. Okay, people, now we're going to look at the economic uses of plants. And I know that you're going to think, we don't need to learn this, it's so simple. But the problem is, you have to remember this, if you understand what I'm saying. You know, there's a lot that you know about what we've studied here, and a lot of it just makes sense. It's just to make your brains aware of that's the way you need to think. So we're taking your information and we're opening our little filing system and we're sticking it in there, into transport in plants, okay, and into economic and structure of plants, and into the economic uses of plants, because this all forms part of plants. So, economic use of plants. We've got trees and plants provide a source of natural medicines, okay, used by traditional healers. They use roots, they use the bark, they use the leaves and the flowers sometimes, okay, to produce traditional cures. Now we call those traditional cures muti, and muti is a proper South African term, muti, and we use it all the time. Even when we're going to take stuff that we've bought from a chemist, we say, come, here's some cough muti or headache muti. Muti is medicine of all types of ailments. Here I've got a picture of a sangoma, but it's a very traditional sangoma. A sangoma means healer, and if we translate it directly to English, it means herbal healer. So sangoma doesn't mean someone who's bad and evil. It's somebody who actually knows how to heal people with medicines. And I mean, there are lots of things. Eating the bark of a fever tree, for example, if you chew it, it will relieve a headache. Using citronella oil in a candle will prevent mosquitoes biting you. So we've got a lot of traditional herbs and things that we use in our everyday life, which has been passed on from generation to generation. And that's what makes our traditional healers so special, is this traditional leader a traditional healer will mentor a whole bunch of other traditional healers and teach them what he knows. So it's passed down from generation to generation. Very South African. Okay, then we have cotton plants produce cotton for the production of fabrics. And here I selected a photo of an actual cotton bud which is about to burst open and release that cotton. Now, cotton pickers will go along and they actually manually pick the cotton out of that little bud, all right, or the pod. It's not a bud, it's a pod. All right, then paper. Now this I found very interesting, is if you take paper and you put it through an electron microscope, that's what paper looks like. And clearly the less gaps you have, the thicker the paper will be. The thinner the paper is, the more little gaps you'll see, but that is what paper looks like. And paper and cardboard are produced from wood, and it gets pulped. That's why we say every time you have this wad of paper, you've actually used a whole tree. You've chopped down a tree. to make that paper. So please, people, don't throw your paper away, recycle it, and get your schools to recycle as well. Old magazines and newspapers, all of that can be recycled at a relatively low cost and prevent the chopping down of yet another tree. All right, so every time you recycle paper, you're saving a tree. Cane and wicker are used to weave baskets and furniture, and I've got a gorgeous basket to show you just now. Then hemp and sisal. Hemp is dacha. Okay, it's a good old dacha, grass, cannabis, hemp. Hemp is one of the species in that family. Okay, and sisal are used to weave ropes. Now, this is a piece of hemp. This is a stem of hemp. And if you unpeel it like that, look at this. This is what they end up making rope out of. And then wood is used for buildings and for furniture. I mean, clearly. This is, look at these bundles. These are bundles of sisal. And that's used to make ropes. So there's your sisal, big bales of sisal. These are houses that are only made of wood. I mean, this whole town is built just with wood. And it's like a fire hazard. But there you've got everything here is wood. Even the window frames are wood. And here's this basket. Now, to put it into perspective here, that's a normal tree. That's a double-story building, and this is this huge big basket that sits in this town. They've got this basket with trees and things growing inside the basket. I think it's actually quite cute. So there are lots of things we can do with wood, and we need to look after apart from using wood to burn, and then it's a fuel, not a fossil fuel, it's a fuel. We use it to burn things, to make fire. Just remember, at the end of the day, this earth, as we know it, has to be looked after, because we're going to run out of things. We're going to run out of places to even grow trees. And if we don't have trees and we don't have plants, we don't have photosynthesis, which means we have no way of converting our carbon dioxide for gaseous exchange, our carbon dioxide to the oxygen, because that's what plants do for us. So respect them, love them, and understand that where we can, we must recycle. All right, now, just to recap, we have a few minutes. Remember, the xylem vessels are there to transport water up a plant. Phloem transports water up and down the plant, but not just water. Dissolved in that water, you're going to find mineral salts that the plant needs. In the roots, the stems, and the leaves, you're also going to find the nutrients that were produced by the leaves. Structure versus function, very important. You must know the structure versus function of the root, and the root hair specifically, because it increases the surface area. Okay? You must know the structure versus function of the xylem tissue. Very important. That we have it's got a thick cell wall, no cross walls. It's just one long, narrow tube. The walls are thickened with lignin. Because that is completely waterproof, we have holes called border pits to allow for sideways movement of water. The fact that they're long, narrow tubes means we have capillarity, which is the force of cohesion and adhesion of water molecules. Okay, all of that. The fact that the xylem vessels are rounded, what does that tell us? They're cylindrical, they're rounded. Why? It gives strength. We've got the sclerenchyma tissue to give strength and support. And all of that is very important, especially in your herbaceous stems, which are your green stems that haven't actually grown into or had secondary thickening take place. When you count your annual rings, remember, annual rings is made up of autumn wood and spring wood. Spring wood is normally wider because that's the main growth period of a tree is spring and summer. Also mainly because in most areas in the world it rains during summer. All right, so it's going to be the time when there's lots of water in the soil and the water has to be carried up. When we look at water transport up a plant, we have root pressure, capillarity, cohesion and adhesion forces, and then transpiration pull is our third process, and it is the most important one because it takes water from a meter up above the ground in a really big tree all the way up to the leaves that are 20, 30 meters high. So it is the strongest force. It's that suction force of transpiration. Make sure you remember the structural changes. So in other words, leaves you'd have to have leaves that are thin, would lose less water than leaves that are large, etc. Also, please people, understand the rate of transpiration and the factors that affect it. And that's going to be humidity, it's going to be light, it's going to be wind, it's going to be temperature, and then, of course, the amount of water that's in the soil, because that water that gets lost must be replaced. Very, very nice essay question. So if I was setting your papers, I would ask you about those 3 processes to make sure we get water up a plant. So learn hard, study well, and make sure you know your plant tissue, because it's easy to learn. All right? Have a good one. Bye-bye.